Hello everyone, I'm Benpeng from Huazhong University of Science and Technology in China. It's my honor to be here and give a talk. The topic of my presentation is Piezoelectric Ultrasound Energy Harvesting Device for Deep Brain Stimulation and Analgesia Applications. This is the outline of my talk. Deep brain stimulation is an important tool in clinical treatment. We can call it DBS for short. This is the precise definition of DBS. DBS is an elective surgical procedure in which electrodes are implanted in specific areas of the brain to control and prevent abnormal brain activity. DBS is widely used to improve the quality of life in patients with diseases such as Parkinson's disease, Alzheimer's disease, epilepsy, essential trauma, addictions, and so on. In this research, we focus on the DBS treatment for pain relief application. As we will learn, for DBS technology, the power supply is the key point. Traditionally, an implantable pulse generator is placed under the skin to address the power supply issue. However, such devices have some disadvantages. For example, percutaneous wires are cumbersome and prone to infection. Additionally, the implanted battery must be replaced regularly because of its limited energy capacity, which brings post-operative pain and financial burdens to patients. So there is an urgent need to develop wireless battery-free passive devices for DBS application. Nowadays, as an emergent technology of wireless energy transmission, piezoelectric ultrasound energy harvesting (PUEH) has attracted more and more attention. As you can see, piezoelectric material can be driven by ultrasound to achieve electric output. So, this kind of material can act like a battery. Most importantly, ultrasound has so many advantages, such as strong penetration, easy concentration of energy, good directivity, and biological safety. According to the regulation of FDA, the safety threshold of ultrasound in human body is 720 milliwatt per square centimeter, which is dozens of times greater than that of radio waves. The safety threshold of radio wave in human body is only 10 megawatt per square centimeter. Here, we will introduce a wireless, battery-free, and passive PUEH device. This ultrasound-driven device is implantable. The aim of our study is to use this device for deep brain stimulation and analgesia applications. According to the piezoelectric ultrasound energy harvesting theory, for a plate in thickness stretch mode, the output power of the device can be expressed as a formula given here. K33 is the electromechanical coupling coefficient, which is changed from 0 to 1. So, this part is a monotonic function. Therefore, for the purpose of increasing the output power of the device. The first thing you should do is to select the piezoelectric materials with large dielectric constant and large electromechanical coupling coefficient. And then you should make this material work at the resonant frequency. As we have analyzed before, for the device fabrication, the material selection is very important. Compared with other piezoelectric materials, samarium-doped PMMPT single crystal owns the largest dielectric constant and the largest electromechanical coupling coefficient. So we selected this kind of material, and the resonant frequency of this material is set to be 1 MHz. To further increase the output power of the device, we design a 6x6 array structure. Additionally, to facilitate device implantation in vivo, the flexibility of the device is achieved through stretchable electrodes and PDMS package. For each discussion later, we named the as developed device SMPUEH device. This is the flowchart of SMPUEH device fabrication process. Firstly, this single crystal with the desired thickness is cut into small pieces, and then 36 elements are collected by stretchable electrodes and packaged in PDMS. For DBS application, a rectifier bridge and a stimulating electrode are integrated. The abatement device, as you can see, is flexible and miniature. The bending angle of our device can be greater than 30 degrees. This device is 13.5 mm long, 9.6 mm wide, and 2.1 mm thick. The total weight of our device is only 0.78 gram. The SMPUEH device has excellent electro performance, driven by 1 MHz ultrasound with the intensity of 2.5 micropower. The maximum power density of our device is 1.1 watt per square centimeter, and its average charging power is 4,270 nanowatt. It's worthy noting that both of these values are the highest results reported so far. As you can see, when the ultrasound is applied using our device, one LED can be lightened immediately. For the Analgesia animal experiment, the formerly induced pain animal model of rats is established. The stimulation target is PAG, which is involved in the pain inhibition. This is the schematic diagram of our experiment. Formalin solution is injected into the paw of the rat to induce the pain. The SMPUEH device is implanted in the brain of the rat and is driven by the ultrasound to stimulate the PAG area for the purpose of pain relief. In order to judge the analgesic effect, the local feed potential signals are collected in the spinal cord. In our experiment, 1 MHz ultrasound stimulation lasts for 5 minutes from the 30th to 35th minute after formalin injection. There are electrophysiology results. It's clear that for the control group, because of no ultrasound-induced stimulation, the LFP signals have no change. But for the stimulation group, during the ultrasound stimulation time, the single intensity is significantly reduced, indicating that LFP signals in the spinal cord are inhibited. These promising results demonstrate that the achievement of DBS and analgesia can be realized by the SMPUEH device. In order to further verify the analgesic application effect of our device, we conduct animal behavioral experiments. Being implanted with the device, the rat can recover after 10 days. Same as the electrophysiology experiment, formalin solution is injected into the paw of the rat to induce the pain. And then DBS treatment is carried out between the 30th to 35th minute. Different from the electrophysiology experiment, the duration of different behaviors of rats are recorded in the whole process, around 60 minutes. Commonly, there are 3 main behaviors of rats after formalin injection. The first behavior is pull down. It means there's no pain. The second one is pull up, which means pain occurs. The last one is pull linking, which indicates the pain has intensified. The time of these 3 behaviors are recorded, and the analgesic effect is analyzed by the formula of the pain score. In the formula, T1 is the time for pull down, T2 is the time for pull up, and T3 is the time for pull linking. It's easy to find that after DBS treatment induced by our device, the pain score is low. But if there is no treatment, the pain score remains high. There are the videos of animal behavior experiments. For the control group on the left, no ultrasound is applied, and the rat keeps linking its paw because it feels very painful. But for the stimulation group on the right, when the ultrasound is applied and the rat feels good. The reason for this phenomenon is easy to understand. When the DBS treatment is induced by our device, the pain is suppressed. Conclusions. Based on high-performance samarium-doped PMMPT single crystal, an implantable PUEH device with high power output has been successfully fabricated. Using this ultrasound-driven implantable device, deep brain stimulation for pain relief has been achieved. This study provides new insights into the development of implantable devices in the future. The details of this research have been published on Science Advances. Thanks for my cooperators and funding support. Thanks for your attention.